Yeah, if somebody is absent, they can go back to the UI capture and we'll kind of go over the slide. So then I'll be talking about specification of motor neuron identities and dorsoventral patterning in the vertebrate neural tube. So these are the highlights we'll be covering in 2 classes. So without the further delaying this, we'll go to the next slide. So yeah. So here you do not have to remember all these kind of things which are mentioned here, but I think if you go over the slide, you can able to remember some of those, because in the exam we are going to put the multiple choice questions, so you can choose one of the answer among the 4 or 5. So so these are the ligands that I have mentioned here, and these are the receptor which the ligand binds to this receptor. So you have the neurotropins ligands bind to this tyrosine kinase family receptor, and you have in the neurotropins family you have the NGF, you have BD, NF, all these things are there. So you just go over the slide and you will come to know which ligand binds to which receptor. So this is neurotropin for the tyrosine kinase. You have FGF family, the fibroblast growth factor family, FGF receptor, HEZOC family, page-range smoothen receptor. These are the targets. You have TGF beta family. You have the TGF beta or BMP receptor. Then you have netrins. SILT. You have the roboreceptor. So all these things are in the slide. So just go over this and we'll put the multiple choice question. Don't have to memorize anything. And then another, so more example of cell-to-cell interaction is also important in the neuronal development. And these are some of the ligands and receptor for cell-to-cell interaction. You have the cell adhesion molecule, the CAM. So you have the receptor, CAMS receptor, delta, you have the notch receptor, afrin receptor. These are the receptor. These are called as receptor protein tyrosine kinase. So another thing that you guys should remember about the extracellular matrix is also important during neuronal development. So the integrin plays an important role for this for development of nervous system. So we'll be also highlighting in the next section when I'll be talking about in the next class. So here I am highlighting some of these signal that is I have highlighted as like last class, like FGF signaling pathway. This is required for the anterior posterior patterning. So the FGF ligand bind to this FGF receptor. So you have the receptor. So this is the extracellular matrix. So it binds to this. Here again, you don't have to remember all the signaling cascade. Just we are going to put the question like what are the signal required for the names of the signal for anterior posterior patterning or dorsoventral patterning, something like that. You don't have to remember all these molecules which are here, but just kind of get an overview of this. So it binds to the tyrosine kinase receptor. Then it activates the RAS signaling pathway. RAS signal activate the MAP kinase. Then MAP kinase activate the transcription factor, CRAVE transcription factor, CRAV transcription factor, which can bind to the, which can translocate it to the nucleus and bind to the DNA, and it express the target genes for the anterior posterior patterning. So this is for the FGF signaling, and this is the key enzyme or key kinases, which you guys should remember a bit. And this is either the FGF bind directly to the tyrosine receptor kinase, or it bind the neurotropins can also bind to this to GRB2, and it can initiate RAS signaling. So this is just to get an overview of that signaling pathway for the FGF. In the next slide, the Wnt signaling I talked about last time. It is required for the anterior posterior patterning, polarity. So there are 2 kinds of Wnt signaling people have discussed or people have studied. One is the canonical pathway. One is the non-canonical pathway. The non-canonical pathway, actually, it is beta-catenin independent. And there are specific ligands for the non-canonical pathway, like Wnt5b, Wnt11. Those are the non-canonical ligands for the Wnt non-canonical signaling pathway. So this is, and here, what happens here is the Wnt ligand bind to this receptor, Wnt receptor, that is the Frizzled receptor, or it can also bind to the orphan receptor, the tyrosine kinase receptor. and it initiates the downstream signaling cascade, which actually, because of the downstream signaling cascade, you have an increase of intracellular calcium. And when you have increase of intracellular calcium, it does so many things. It can also, like activates the calcineurin, which is a calcium regulator, and also it activates the CAM kinase 2. And the CAM kinase 2 or calcineurin further, activate NF-kappa B or NF-AT, which translocate to the nucleus and bind to the target gene and transcribe the genes required for the anterior posterior patterning during non-canonical Wnt signaling pathway. Or it can also, the orphan receptor tyrosine kinase can also activate CDC42 row kinases, which can activate AP1 or AP transcription factor, and it can translocate to the nucleus and also initiate the expression of target genes. So this is the non-canonical mediated beta-catenin independent Wnt signaling pathway. And the ligand and those ligands for non-canonical signaling pathway, I have not mentioned in any of the slides. These are Wnt5b or Wnt11. You can note it down. So these are the non-canonical ligands. Now we'll go for the canonical one, which is the beta-catenin dependent. So the Wnt molecule bind to the Frizzle receptor, and it activates this complex that is called as Drosophila complex, which can further activate beta-catenin, and beta-catenin translocate to the nucleus. It is complex with TCF or LEF, and again bind to the DNA and express the target gene. So this is just this is another signal required for the anterior posterior patterning during development. Now, another is the retinoic acid, as I mentioned, it required for also for the anterior posterior patterning. So these are the different signals that just kind of give a overview to to show that these are important signal for the animal development. So retinoic acid bind to the retinoic acid receptor, and after binding, it translocate to the nucleus. The retinoic acid receptor also translocate to the nucleus. Along with retinoic acid, it bind to the DNA and again initiate the expression of target genes. Another ligand is the BMP. This is so those ligands that I discussed before, like Wnt ligands or FGF or retinoic acid. Those are required for the anterior posterior patterning. BMP or sonic hedgehog. These are required for the dorsoventral patterning. So you have anterior, posterior, you have dorsal ventral. So those signals are required for the development during development. So anterior, posterior, dorsal, ventral. Got it? So yeah. During the development, it happens. Yeah. So you have some signal. So it acts like a morphogen. Suppose you have the Wnt signal that is required for the anterior posterior patterning. So anterior mostly the levels will be higher in one side and lower in the another side during anterior posterior axis. Similarly, in the dorsal side or ventral side, you have the gradient. So it happens at the same time. Yeah. So different signal activate, and so that you will have development. Yeah. So this is the BMP. This is more studied ligand, the TJ beta family, required for the dorsoventral patterning. So BMP molecule bind to the BMP receptor. So these are receptor serine kinase, and these required for the generation of the epidermis. And basically noggin and codin, as I mentioned, they require for the neural induction, and BMP inhibits neural induction. It actually shut down neural induction. It initiates the formation of epidermis. So when BMP bind to the BMP receptor, the receptor serine kinase, you have activation of you have formation of a SMART, and then it make complex with this co-SMART. It activates SMART, and the SMART again make a complex with co-SMART, and that complex translocate to the nucleus and again transcribe the genes. So and when this thing happens, this is required for the formation of the epidermis, and this actually noggin and codin actually required for the neural induction. So when the neural induction happens, the BMP comes and shut down neural induction because it make a complex with gain encoding. So the inhibitor for the neural induction is the BMP. So we'll go to the last signaling molecule. That is sonic hedgehog, is also required for the dorsoventral patterning. And these are the receptor for sonic hedgehog ligand, which is patched protein, and you have smoothened. So they form a complex receptor. And when this binds to this receptor and it activate GLI1 transcription factor, I'm forgetting the full name of this. So I think glioblastoma-like transcription factor, something like that. So it binds, it translocates to the nucleus and again, initiate the expression of target genes. So during anterior posterior patterning, you have activation of a certain signaling molecule like Wnt signaling molecule, you have activation of retinoic acid, and dorsoventral patterning, you have activation of either BMP or sonic hedgehog. So we'll be talking more about this when I will take the case study for the Drosophila. So any question in this section, because we are going to jump to the next section about Drosophila development and anterior posterior patterning of Drosophila. So these are the signaling molecules to remember that which is required for the anterior patterning, posterior patterning, dorsal patterning, or dorsoventral patterning. So I think 4 or 5 times you will go over the slide, you will remember. But I will not going to put much question on this section because these are something not required for the analysis. Like you cannot sometimes you'll forget. So I will not put much question of this, but it is good to remember those things. Okay, so if there is no question, we'll go to the next section. Oh, why this is not moving? Okay, so we finished with the first part, which is the important signaling molecule involved in patterning. So now I'll be talking about the well-studied model, that is Drosophila, and how dorsoventral patterning happens in the early embryo during neuroectoderm formation. So I'm using these model organisms. I'm a Drosophila neurobiologist from last 11 years. So I have every day I'm seeing the flies. So flies are very beautiful, and you have like humans, you have a lot of the genes are conserved in flies, which you can study neurodevelopmental disease, different aspect of neural development. So like humans, you have two different sex, like female fly, you have male fly, and the developmental cycle or generation of Drosophila, the life cycle of Drosophila, from one generation to another generation depends on what temperature you are growing. You can grow this Drosophila in different temperature. Like you can grow this in 18 degrees centigrade, you will get a delayed development. It takes around 12 days to get animals. If you grow it 29 degrees centigrade, you will get 5 days to get animals. If you grow, this is 25 degrees, so it takes around 7 days to grow animals because this is the room temperature. So these Drosophila, after mating, they lay egg and develop into embryo. A lot of things happens during embryo development. Then after embryo, you have development of larvae. So there are different sets of larvae, like first instar larvae, the smaller larvae. You have second instar larvae, little bigger, and the third instar, which is people are using as a model system to understand different aspects of neurobiology. And after third instar, it takes around like 1 day to first instar, second instar, another day, third instar, another day. So it takes around 3 days to get the third instar larvae, fully grown larvae, and another 2.5 days to 3 days take to become an immobile state. Like those are mobile, they can move. And this is the pupa stage, so they are remain immobile, and this is called as pupa, and takes around 3 days. And from pupa to adults takes around 2 to 3 days. This is very simple, and there are a lot of things happens during development. So we'll be talking about like how the development happens in Drosophila. And before going details to the Drosophila development, I just want to highlight some of the structure that you can map, and which structure forms what during during development of Drosophila. So there are certain vital dyes which are people have used from the past to level different group of cell in Drosophila. So these are the imaginal tissue which can develop into future structure. So there are dyes to identify eyes in the embryo, or the proboscis where he takes food, or the wing, or the abdomen, or the neural tubes. So in mammals you have dorsal neural tube in the dorsal side of the animal. You have the nervous system. In the invertebrates like Drosophila, you have in the ventral side the ventral neural tube. So let us talk about how gastrulation happens during development. Last class, I talked about gastrulation in frog in the dorsal blastopore lip, and also I talked about the primitive streak in the chicken. So similar, and these are called as spemen organizer. So you have something like called spemen organizer in Drosophila, and here the gastrulation happens. So these are the different steps of gastrulation. So you have the accumulation of the dorsal protein, and this is the ventral side, and this is the dorsal side. So you have accumulation of certain protein in the dorsal and ventral side of the during the development of the Drosophila embryo. So you have accumulation of DPP, the decapentaplegic protein in the dorsal side. In the ventral side, you have the sonic hedgehog that I talked about in the last slide, sonic hedgehog. And you have also accumulation of, these are the short gastrulation protein. And after that, you have, formation of certain structure called as dorsal ectoderm or ventral ectoderm or a neuroectoderm. And also you have initiation of the mesoderm. So the mesoderm actually invaginates inside at the junction of neuroectoderm. So here you can see that the mesoderm invaginates inside at the junction of neuroectoderm, which forms the nervous system, the neuroectoderm. Now you have the you have the three distinct layer, like frog. I talked about ectoderm, endoderm, mesoderm. And because of invagination, you have this formation of three distinct layer. You have outer side the ectoderm, the middle side the middle you have the mesoderm, then you have the endoderm. And eventually you have formation of musculature, you have intestine, and you have the, from this structure, the neuroectoderm, you have formation of the ventral ganglia. So this is how the, gastrulation happens in Drosophila. And here some of the proteins, just I want to highlight again. The dorsal side, you have the DPP mRNA or DPP protein accumulate in the dorsal side. You have the sonic hedgehog in the ventral side. Okay, so these are also mentioned here. And as I mentioned in the last slide, yeah, so Drosophila have, like in the amphibians, you have dorsal blastopore lip. In the primitive streak, in the cheek, you have the primitive streak. Similar, you have Drosophila spemen organizer, you have here. So so like in the junction of neuroectoderm, the mesoderm invaginates inside. So that is called as spemen organizer, where you have invagination and formation of three distinct layers. So and a lot of cell migration and it happens during the formation of mesoderm. So these are the proteins that you have to remember, that DPP protein accumulate in the dorsal side and sonic hedgehog in the ventral side. And these are the signal highlighted. So the DPP, dorsal protein, sonic hedgehog, the ventral protein. And here again, just want to show again the neural induction in Drosophila. So the you have this noggin coordinate polystatin. It activates neural induction, okay? And the BMP, it inhibits. So when you have the binding of BMP with this protein, noggin coordinate polystatin, and this cannot bind to this receptor, so you have shutdown of neural induction. But when you don't have this BMP, this noggin coordinate polystatin, they bind to this receptor. Now you have the formation of neural tissue. And similarly, like DPP and sonic hedgehog, it also happens. Another type of BMP receptor called as thick vein and pont. So the sonic hedgehog binds to the pont, and it induces neural tube formation. and DPP, another protein. Actually, it accumulates in the dorsal side. So it complex with the sonic hedgehog, so that sonic hedgehog could not able to bind to the pond. So this is how you have regulation of generation of neural tube or develop morphogenesis during development of the Drosophila of different tissue. So this is just overview. These are the receptor I have just highlighted, kind of things. Here I'm comparing between vertebrate and Drosophila. So in Drosophila, in the dorsal side you have DPP, and the ventral side you have sonic hedgehog, and this is how the initiation of the neural ectoderm happens in the ventral side of the Drosophila. But this is opposite in vertebrate. It happens in the like in the dorsal side, the caudine, noggin, and polystyrene. It happens in the dorsal side, and in the Drosophila it happens in the ventral side. Just to compare about how neural induction happens in vertebrates and invertebrates. So any question in this section? Next, before moving to the next section about specific experiment people have done. Okay, so if there is no question, then we'll go to the next section where Lewis and Bollard, they have done genetic studies in Drosophila to kind of put a hypothesis that how the development happens, what are the molecular mechanism of anterior posterior patterning during Drosophila embryo development. So three like Lewis, Christian, and Eric, they have done work for the anterior posterior patterning in Drosophila, and they got Nobel Prize in 1995. So here, before going details about the developmental the work they have done, so here I just want to highlight these are different structure that present in Drosophila, and you have the head, and this is the embryonic stage, and this is adult stage, and you have there are three distinct components in adult Drosophila. One is head, another is thorax, and the last part is the abdomen. So in the head, you have this is the head, and the thorax you have three different segments like T1, T2, and T3. T1 is the prothorax, and T2 is the mesothorax, and T3 is the metathorax. And now the third part is the abdomen. So in the abdomen, it is the arthropod is segmented. So they have worked, the Eric and Christian and Lewis, they have worked like what are the genes required for the segmentation in the Drosophila. So in the abdomen, you have 8 different segments. Starts from A1, abdominal 1, abdominal 2, up to abdominal 8. So these are also same structure are also present in the Drosophila embryo, and this is the Drosophila adult. So now they have done several studies, and they have identified several mutants which is required for the dorsoventral like patterning in the Drosophila, and they have identified some of the genes called as homeotic genes that affect segmental identity. So during earlier days, Lewis he has identified. So this is the normal wild type Drosophila. You have the head here, you have the thorax here, you have this abdomen here, and you have the heart here. Like in human, you have the semicircular canal in the ear that act as a balancing organ. Like in the flies, during they fly, the halter actually these are rudimentary wing. They helps in the balancing, keeping the proper posture in the during flying in the Drosophila. So Lewis identified that there is a set of gene called as the homeotic gene called as ultra-wide thorax. So when you have mutation in the ultra-wide thorax, it develops two thorax. See, one thorax, another thorax. Here is only one thorax. So you have two pair of wings, present in this in the species of Drosophila mutant fly. So it affects the segmentation, because normal segmentation it doesn't follow the normal segmentation. So normal segmentation, you have a head, you have a thorax, you have the abdomen. In thorax, you have T1, T2, T3. In the abdomen, you have A1 to A8, or sometimes A9. So they found that because of the mutation of ultra-bithorax, the homeotic mutations. So I will be talking about homeotic gene in the next coming slide. So they found that you have two double thorax and you have a double pair of wing. So now you have another set of mutation called as antinopedia. So this is the head of the Drosophila, And here these are called as antenna. Here this is another one antenna, another antenna. So they help in the olfaction, smelling, olfaction in the fly. So when there is mutation of antennopedia gene, the leg is converted into antenna. So they have identified that this is another homeotic genes, segmental genes, required for the body segments, formation of the body segments. So for this work, he got the Nobel Prize. And similar kind of thing also exists. Not 100% happens what is happening in Drosophila, but similar kind of thing also exists in vertebrate during vertebrate nervous system or vertebrate development. So we'll be talking more about that in the next class. So here these are the mutants. So this is mutation in the embryo, sorry, in the larvae stage. These are the mutants, and this is the wild type. So in the wild type, you can see you have this structure. These are called as trachea. The trachea are present in the wild type, but they are missing in the mutants. So they have identified there are a set of genes. The homeotic genes is also required for the development in Drosophila. So this is all about the Lewis work. Then I'll be talking about how anterior posterior patterning happens in Drosophila and what are the genes that are required for the anterior and posterior patternings. So you have three distinct set of genes. One is cytoplasmic one is some of the genes that are carried out from the maternal the mother Drosophila. These are called as maternal genes. They actually kind of helps in cytoplasmic polarity. You have the gap genes, then you have the pair genes, then you have the pair genes, again you have the segment polarity genes and homeotic genes. Just before I was talking about the homeotic genes like ultrabithorax or the antennopedia. So first, when there is fusion when there is development of Drosophila egg, so you have the sperm nucleus and egg nucleus. So after fusion, so you have some of the genes, for the development. So some of the genes that are carried out from the female Drosophila, these are called as maternal genes, and that define the cytoplasmic polarity. So after during the development of embryo, you don't have any transcription of genes. So and the cytoplasmic or the maternal genes, they actually helps in the maintain the polarity in the embryo. Then after some time, like after cellular, like you have fusion of the nucleus, then you have formation of you have division of the nucleus, then you have kind of a multinucleate structure called a syncytial stage. Then you have a cellularization stage. Then after that, you have expression of genes. Those are the gap genes. So the maternal gene activate the gap genes. Now, when you have expression of gap genes, it activates the pair genes. Now the pair gene again activates segment polarity gene and homeotic genes. So it's a cascade. So it starts from the maternal gene, then it activates gap gene activates pair gene, and the segment polarity gene and homeotic genes. So the expression levels of one gene, kind of activates the next set of genes. Okay, so these are the segment polarity genes. These are the gooseberry genes or the patch genes. The pair genes is even skipped or skipped, not paired, and the gap genes are crupule or mute. So this you just have to kind of go over the slide to remember some of the genes for the gap, pair or segment polarity. So any questions from this section? Okay, simple, right? It is Drosophila, that's why. You have complicated thing next class. So because yeah. So I just want to go over how this thing, this development happens, like polarity, cytoplasmic polarity, or how the maternal genes, then how it activates the next set of genes, the gap genes or the pair genes, and the segment polarity and homeotic genes. So this is the first step of the embryo development in Drosophila, and you have the nucleus. After fusion, it forms it forms the male nucleus fused with female nucleus and form nucleus. Then that divides into further divides into forming two stages, 2 nucleus and 4 nucleus, 6 nucleus, like that. Then you became and it became mass of the nucleus in one particular compartment, and this is called as syncytium, and this is the anterior stage, and this is the posterior portion of the Drosophila embryo. So no embryonic transcription before cellularization. So here there is no transcription. Okay. Yes. Yeah. Yeah, those are the different sets of genes that are expressed during development. So I'm just going over the beginning, like maternal genes. So we'll be coming to that. So now, after formation of a syncytium, so you have some of these transcription factor that is for the maternal transcription factor or maternal transcription, like the mRNA for the maternal transcription factor. These are carried out during from the mother, and what happens these transcription factor, they kind of anchor to either anterior side or posterior side of the embryo, like either here or here. So you have this anchor protein that is also carried out from the mother. So then what happens is, so here this is the RNA in situ hybridization to show some of these maternal genes that is being kind of carried out from the mother. One is the B-cod genes, which actually act as a morphogen. So this is the B-cod genes. The B-cod genes, it is important genes that is required during the oogenesis in the female fly. So you can see that you have more accumulation of the B-cod genes in the anterior portion of the Drosophila, and this is the B-cod mRNA. So people have done the RNA in situ hybridization to visualize this in the embryo, or they have done antibody staining or GAP tagged construct to visualize the B-cod mRNA in B-cod protein in the anterior portion of the during the development. So B-cod actually accumulate in the anterior portion. This is a maternal gene, okay, transported carried out during the development from the mother. So again, this is another technique that if you express B-cod under the promoter GFP, if you express a fusion protein with B-cod with GFP, you can also visualize that with RNA in situ hybridization. You can also visualize that in the it is accumulated in the anterior portion during the development. So B-cod specifies anterior polarity. Okay, so here some of the experiment people have done that it is required for the anterior development. So this is a wild type Drosophila. With RNA in situ hybridization, they found that the B-cod expressed in the anterior portion of the Drosophila because it specifies head. And head, now you have the tail this side, the posterior side. So if you have mutation in the B-cod minus, you are missing this head. So that is how the genes are identified, which are anterior, which genes are expressed in the anterior side, which genes are expressed in the posterior side. Okay. Now another study. This is a B-cod mutant. Okay. And you have no development of so if I coming to the previous slide, so B-cod mutant, there is tail in either side, no head. So if you inject B-cod mRNA in the anterior side here, same thing they have done, you can see that there is development of head in this mutant, which suggests that B-cod is an anterior specific gene. Okay. Now, if you inject the B-cod mRNA in the middle, it develops head in the middle. So here you can see the head is there, tail in either side. Okay. So if you inject B-cod mRNA in the posterior side, you have development of head in either side, two heads. Here is the middle head, here is the normal head. Okay. Very interesting, right? So this kind of experiment, suggests that B-cod is B-cod specified, anterior polarity. Now, there are set of genes or mRNA which is also expressed in the posterior side to define posterior polarity, which is the one of the mRNA that is expressed in the posterior side is the caudal. So you can see that there is it acts as a gradient, like a morphogen. You have higher in one side, lower in another side. You have a higher in one side, lower in another side. So, becoid express more in the anterior person and less in the posterior person. And the caudal express more in the posterior person and less in the anterior person. So this is how you have polarity by expression of either becoid or caudal, and that is how the development happens in Drosophila. So after so here again the formation of syncytium. So there are other set of genes also expressed during development, that is nanos. So that also expressed in the posterior side, defines posterior portion of the Drosophila. So anterior side you have expression of becoid, develops head. Then so these are the things like specifies anterior posterior polarity in Drosophila. So here again these are the anterior specific gene, that is becoid or hunchback, and you have caudal and nanos. These are the posterior specificity, define the posterior specificity, and these are the maternal gene products, carried out from the mother, from the female Drosophila. So here is the graph showing the expression of different mRNA and in different stages of the development. This is the oocyte mRNA. This is a oocyte. This is in the oocyte stage, and this is in the early embryo stage. In the oocyte stage, you have higher expression of becoid. In the anterior side, you have a higher expression of nanog. In the posterior side, and you kind of you hunchback and caudal. There is no change in the expression. And when it develops into early embryo, and there are a lot of things happens. So you have higher expression of becoid. It goes down in the posterior side. You have a higher expression of nanos in the posterior side. It goes down in the anterior side. And also you have higher expression of hunchback in the anterior side, and higher expression of caudal in the posterior side. So this is during the early embryo stage, and this is during the oocyte before the cleavage. Okay. So after cellularization, you have transcription, formation of new sets of gene, transcription of new sets of gene. So that is called a zygotic transcription. Before that, I talked about the maternal things. Okay. So these are summarized here. Some of these things in the zygotic stage. So becoid and nanos, these are maternal genes. Okay. So this is just a highlight. So if you go over this slide, you can actually remember some of the things which are expressed in the maternal, which genes are carried out from the maternal animals, which are actually transcribed during during zygote development. Those are called zygotic transcription. So you have the initial transcription of the gap genes, that is crupple, and you have expression of parul genes, that is even skipped, and also the segment polarity genes like engrilled or wingless, and the homeotic gene, the Hox genes, that is ultrabithorax and abdominal A. So that actually by the action of all the genes, you have the development of a Drosophila, which has a head, thorax, and abdomen. Okay. So same thing kind of summarized here. This is the earlier genes. Earlier genes are nanos, becoid. Then the intermediate stage, you have the giant crupule, the parul gene. Then you have the latter, you have the segment polarity gene or homeotic genes. So this is just to kind of revise again. Now we'll come to the segmentation in Drosophila. Human doesn't have this, or animal doesn't have this kind of segmentation. The insects, they have segmentation. They have the body is divided into 3 different segments in Drosophila. One is the head, like thorax and abdomen. Again, each segment is again subdivided into other segments. Like head is divided into 3 like one segment. Head is, there is a different person in the head. You have, you have labium, you have maxilla, maxillary segments. You have labial segment. This is in the head. You have in the thorax. You have T1, T2, and T3 segments. In the abdomen, you have again A9, A2, A9 segments. So why to discuss segmentation? It plays only small role in vertebral neural development. Small role. It's not zero, right? It's not zero. There is some role. Some of the genes is also required for the vertebrate segmentation. That's why it's kind of important to study, this thing in the lower animals. That's why I'm just kind of covering some of the genes required for the segmentations. So here, this is a Drosophila embryo. Again, you have these are the different segments. So which are like mentioned here: maxillary segment, labial segment, thoracic segment, and abdominal segments. Again, each segment like T1 segment, again, each segment is divided into compartments. Like you have two compartments in maxillary segment, like posterior compartment and anterior compartment. Each segment is divided into compartment, posterior, anterior, and there are a set of genes which is also expressed, which also defines posterior compartment, anterior compartment. And each compartment again is a kind of grouped into parasegments. So you have 14 parasegments, and these are the genes which define parasegments, which is I have written here: FTZ, which is Fuji Taraju, and the even skipped. It's a very weird name, right? So we will be going slowly on this. So the segment polarity genes define the anterior posterior borders, okay? And the gene expression boundary in the embryo are defined by parasegments. So don't mug up anything, just kind of remember that the insect body is highly segmented, and those are again regulated by the different sets of gene. Okay, so we'll go to the next thing, the parasegments. Okay, so this is these are the different segments, like 1, 2, 3, 4 segments. So I've just kind of snapshot here 4 segments, and each segment is again divided into parasegments. And these are the set of genes which are expressed in the parasegment, which is even skipped or FTJ, that is I mentioned before. What is that called? Fuji Taraju. Okay, so you have more expression of even skipped in the anterior side and less expression in the posterior side. So this is anterior, this is posterior. Again, anterior, posterior, anterior, posterior. So this is how you have, because of the expression of EV or FTJ, you have segmentation of the body. Okay, so you have more expression of EV, even skipped in the anterior side. You have less expression in the posterior side. Again, more expression. This is the anterior, this is the posterior, like that. So, and if you go more in the further detail, you will find that some of the other genes which also specifies anterior posterior polarity. That is, anterior polarity, that is engrilled. It specifies the anterior polarity, or the wingless, it specifies the posterior polarity. So you have repetition like this in each segment, like anterior polarity, posterior polarity, one segment. Another segment, engrilled, anterior, posterior. So another. So this is kind of a repetition in each segment. Okay, engrilled defines the anterior polarity, wingless defines the posterior polarity. So, or another anterior polarity gene is a hedgehog, like engrilled. Okay, so clear. Different set of genes defines anterior and posterior. Anterior polarity is defined by even skipped. Posterior polarity defined by FTZ. Okay. Or anterior polarity engrilled or hedgehog, posterior polarity wingless. So just highlight. So then the Lewis model for specification of a segment identity. So as I mentioned before, you have different segments like head. This is thorax. This is maxillary segment. This is the thoracic segment. This is abdominal segments. So here, what Lewis has done, so he has mutated several genes and kind of know. He kind of want to understand which gene is required for the anterior development, posterior development, or different segment, head segment, abdominal segment, or thoracic segment. So he has done this kind of study, and for that he got Nobel Prize. So here, different set of genes. So wild type, the dark circle is called. These are the genes in the wild type genes. So some of the genes he mutated, and he called that a strongly reduced gene. And some of the genes is also not essential for the polarity. So he got a lot of combination. Okay, this gene is, when he reduced this expression of the gene, he found that okay, one segment is missing. One segment, the Drosophila larva is not able to develop like a wild type larva. So this is just kind of to show his experiment and to define segment identity or segment polarity. Now we'll come for the last section of the gene, like pair rule genes activate segment polarity gene and homeobox gene or homeotic genes. These the homeotic genes also present in the vertebrates. So in the Drosophila, you have one Hox gene or homeotic gene, In mammals, you have on the vertebrates, you have four homeotic genes like HOX-A, HOX-B, HOX-C, and HOX-T. So these are present in different chromosomes. HOX-A present in chromosome 7, HOX-B present in chromosome 17, HOX-C present in chromosome 12, and HOX-T present in chromosome 2. So again, in Drosophila, just going back to the lower animals, the Drosophila, the Hox gene cluster is actually present in two clusters. One is called as antenopedia complex and is called as ultrabithorax complex. So this ultrabithorax complex, this defines the thoracic portion of the Drosophila, and this defines the antenopedia complex, defines the anterior portion, the head portion, or the thoracic portion of the Drosophila, and ultrabithorax there defines the posterior portion of the Drosophila. Okay, so and you have similar kind of homology, similar kind of gene also present in the vertebrates. So Hox genes, these are homeobox genes that can form body parts. Okay, like in Drosophila you have antenopedia and bithorax. The bithorax defines the posterior portion like the like the abdominal thoracic portion. Antenopedia defines the anterior portion like the head portion. Okay, and there are certain transcription factor like kind of expresses during this development. So you have for the antenopedia complex, you have set of transcription factor. You don't have to remember those. And for the ultrabithorax, you have a set of transcription factor that defines the body or defines the development of the Drosophila. So so the same thing I've just mentioned about vertebrates, four sets of the Hox genes or segment polarity or homeotic genes in vertebrates. So so this is all about the Drosophila development. So next class we'll be talking about the vertebrate development, so which is more of you guys will be interested. So if you have any questions in Drosophila development, you can email me, or you can discuss more about this if you are interested.
Where are like pictures coming from? Which types of?
Oh, so let me see.
Yeah.
Let me